And Dr. Lukman, if possible, please share your screen. And because I have reset my PC, and so many packages are not available in R. Okay, please share it again. Okay, and today our task is gene annotation. So gene annotation mean map the gene information with database. So different methods for gene annotations are available, but we are working in R, so we will learn according to R. So you can type a definition. Gene annotation mean give hashtag map gene information with database. It tell us about function of gene, what is the role of gene, pathway involved, and its biological role. Okay. Now in R also many methods for gene annotation. So if you have, please give hashtag for gene annotation. If your RNA-seq belong to model organism, if your RNA-seq sample or data belong to model organism, you can use ready-made package for gene annotations. We can use ready-made package for gene annotation. But if we don't have, if our, but if sample, if RNA sample are other than model organism, then we will use some database and REF file. Can say database. Okay. Now today we will work on if our organism belong to model organisms, how we perform gene annotation. So please give hashtag with all lines, line number 2, 3, 5, 6. Okay. Now for gene annotation, you also need gene expression analysis. So it is the next step in RNA-seq analysis. So first step is gene expression analysis. After getting expressed gene, or we need to move gene annotation. So please, all participant open ChatGPT. Please open ChatGPT. ChatGPT. Open new chat. Now write a message. I want to perform gene annotation. Can you give me packages specific for gene annotation for model organisms? For model organisms. It will show you some packages name. If you want to use human dataset, mouse dataset, or other model organisms, you can use these packages. So send this message. Scroll down, scroll down. Okay. Now come on first step. And copy this and share in group so that other participant can also read this information. And share in group. And please read this information. Okay. But a lot of tools are available. So You can read it later. Now we want to perform gene annotation. So please open your R Studio. Take str of your sample info and count data. You have typed wrong spelling for both line number 1 and line number 2. Please just remove these lines. Remove these lines. Type str. Select and type col. Sorry, sample info. Sample info. And select from suggestion. Okay, run this line. Again type str. No, please type a small count. Small counts. And run this line. Okay, copy these two str. Paste here. And write a line at start. I want to perform at start. I want to perform RNA-Seq analysis. Please first check dataset suitable for analysis. If not, rearrange. I want to perform RNA-seq analysis. Sorry, okay. I want to perform RNA-seq analysis in R.
Sir, if not arranged, so then
If not arranged, please arrange for RNA-seq. I want to perform in R. And send message. Please read all instructions from start. Okay, scroll down.
Samyari.
Okay, scroll down. You need to read all these instructions. So please come on first step so that all other participants can also read this. It is showing you your data structure suggests that you have sample information. Sample information means metadata stored in sample info and gene expression count data stored in counts file. So it's saying you have two files. One file is metadata and other file is gene expression count data. So detail of first file is sample info. Correct metadata format, file name, sample name, cell type and status. But in your case, potential issues ensure there are no missing values in sample info. Okay, but in your data, now come on your data, check on small counts. Potential issues, the dataset contain only 10 genes and 4 sample columns, which is too small for standard RNA-seq analysis. Second, small count like row names, genes. You need to gene identifier, Ensembl IDs or gene sample. So please, it is showing you and genes names or IDs are not available. So please open RStudio. Click on count small counts. There is no heading. Okay. No, it's not issue. Again, click on ChatGPT. No, second step is make sure the counts are not normalized. Raw counts are required for differential analysis. So pre-processing or fixing. If small count is a subset, load the full data set, but we have only small data set. I think I have shared you two files instead of airway dataset. These files have large dataset. We will do practice on these files also. Ensure row names are gene IDs. Check for zero count genes, filter them. If there is a zero count gene, you can filter and remove. Verify all sample information, sample names, entries, match column names in small count. So it is very important step. If the sample info names not matched with small counts data, it's mean and there is a issue in your dataset. If you have columns in 10 columns, there should be 10 rows in sample info. If 12 rows, 10 columns, there may be issue. So no, process the data in R by checking for missing values, ensuring gene identifier exists, rearranging small count to match sample info, and saving the clean data set for RNA-seq analysis. Scroll down. Now here is the first step. Cleaned RNA-seq count data. So here are the findings from the initial data check and cleaning. But we want to perform all in R. So please write a message. I want to perform all these steps in R. So here is the R script that will pre-process your RNA-seq data by performing the following step. Load the required libraries, check for missing values, ensure gene identifiers exist, rearrange the count matrix to match sample info, filter low out or low quality count genes. Okay, now scroll down. Here is the sample data, but we already have data set, so we don't need to use this sample data set, but wait until the completion of the script. Okay, copy this code, copy. No. Click on copy code. No, please. Maximize the screen and copy from clipboard. Okay, run these lines. First do library, click on run button. Next. Okay, you have all these packages. Now load sample info data, but you already have data set. So you don't need to run this code. Scroll down. Yes, click on line number 26. You can remove these two codes, above codes, because these are a sample data set, not your original data set. You can remove these. And you already have this data set in screen number 3. Okay, ensure column names match sample info, sample names. Run this line. Yes, run this line. It is showing you error. Name attributes 12 must be the same length as the vector. So it's mean sample info and count data. Gene names are not same. You can check in your screen number 3. Sample info, observation 10, while count data 10, 12 and 10. So we need same. So copy this code with script. No, not complete, just these 2 lines. Copy, paste here, and send message. The error occurred because the number of columns in small count, which should correspond to the sample names, does not match the number of sample name entries in the sample info. So it is the main issue. Your sample info contains 12 samples, but small counts has only 4 samples. So how to fix? Check the dimension of both data frame. Copy this code. Scroll down. Paste here. And run line number 36 and 37. Yes. And open ChatGPT. Next code is ensure that small count has been column name that match. Okay, if small counts contain only a subset of sample, subset sample accordingly. Okay, here you need to provide a result of first script. So open R, copy these results, last line, last 2 lines. No, in screen from screen number 2. From screen number 2. Yes, here are the results. 12, 4, 10, 4. Copy. Yes, paste here and run. Okay, first read instructions. It is showing you your sample info has 12 sample while small count has only 4 sample, which cause the mismatch error. Fix this issue. Since your small count only has 4 samples, we need to subset sample info to only include the match samples. So updates step in R. So next is copy this code, paste here and run. Okay. Now again open ChatGPT. Next is rename the column of small count. Copy and run. Okay, next. Double check that everything match. Okay, copy and run these lines. Okay, next, next, next. Okay. Now again open next step. Okay, say thanks. I have run this code successfully. Now I want to perform next step. Okay, send this message. Okay, next step is filtering low count genes. Remove genes with very low counts across all sample to improve statistical power, creating a RNA-Seq dataset, normalizing the count data, exploratory data analysis, and differential expression analysis. So scroll down. Here are codes for filtering low count genes. Copy, open R, paste here and run. Next, 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 next. Okay. Now again, open ChatGPT. After this step, once you have filtered low count genes, let me know if you want to proceed with yes. Type yes, I want to proceed for next step. Yes, I want to proceed for next step. Please wait. Okay, copy the script. Okay, you can ignore these sample data, and you have already done library. So please ignore first two codes. Ignore them and move to next step. Next, please scroll. Yes, line number 81. Next, 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 next. There is no missing value. Okay, next, 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 next. Okay, now here is the DEGs dataset. Factor level have dropped which had no sample. It means you have dropped some low quality reads, and because our dataset is very small dataset, and this code have removed some data, so factor level were dropped which had no sample. Okay, so it's mean we don't need to move next step. So please clean screen number 1. Sorry, screen number 3. Screen number 3. Okay. Again import these files because your data set have been changed. You can airway counts and sample info. Airway counts, yes. Select yes heading. Okay. Okay, next. Airway sample info. Heading yes. No, it is a big data. Okay. It is suitable for RNA-seq analysis. Okay, take str. You can remove all these codes, select all and delete. Okay, run these two lines. Yes, you can delete from with brush button screen number 2. Okay, now run these lines. Next. Okay, now copy both str. Open new chat. Open new chat. And write a message. I want to perform RNA-seq analysis in R programming. Okay, send this message. Okay, scroll down and copy these codes. Let's do libraries. You need to run library. Don't run this code for installation from line number 10. Next. Next. Okay. Next. There is no package called and okay. You can install by removing command line number 7, 8, line number 8. Type BIOC manager. BIOC manager. Select BIOC manager. Select install button. No. After selecting bioc manager, it will give you suggestion list. Remove again and type install. After bioc manager, type install. No, please. No. Remove. Remove R of manager. Yes, again type. It will give you suggestion. BIOC manager. Select BIOC manager. It will give you suggestion for installation. Please select BIOC manager. Yes, here is the install. Select install. And within this type inverted comma and provide package name. You can copy package name from screen number 2. Give an inverted comma, before and after, and run this line. I think you need to add inverted comma, not tick. Inverted comma. You can copy the name from line number 7 with inverted comma. Last word after feet map. Yes. Okay, now run this line again. Come on, click on session button, screen number 1. Session, click on session, screen number 1. First line at the top of screen number 1. File, edit, code, view, plot, session. Yes, click on session and select restart R. Okay, now run this line again, line number 8. Yes, now it's working. Okay, press A, type A in screen number 2. And yes, type A and press enter button. I think we need to wait more. Okay, please all participant before next class run DEG, DESeq 2 and save the result in your R so that we will continue our class from last step to onward. Okay, please all participants run these codes and also try to perform, but it is showing may take some more time. I think it may take time because files are heavy. So please all participant do practice again on these files, airway counts and airway sample info. And after completing volcano plot, MA plot, heat map plot, PCA plot, you need to save these results and script so that when we start our tomorrow class, we will continue to this step. Okay, thank you. Allah Hafiz.